Hello, in this video I show you how to use the dssp tool of GROMACS package to define a secondary structure of your protein. dssp is arguably the most widespread method for defining secondary structure, which is based on the hydrogen bond patterns of the backbone of your protein. It is not exclusive to GROMACS, and you can find a lot of dssp implementations, including the original one, as open source on the GitHub. In fact, the dssp tool was added to the GROMACS package relatively recently in 2024. dssp tool requires you to provide the path to your trajectory file, topology file, index file, and also to specify dynamic selection of the index group. As for the output, it currently produces two output files. One is the main file, .dat file with structural information, and the second one is the XVG file with statistics which you can plot. Calculations here are pretty fast because I use pre-aligned trajectory with removed boundary conditions, stripped of water molecules and ions. If you don't provide the dynamic selection, it will by default use the whole protein index group, and in this case I have two protein chains, so I want to focus on only one chain. Now let's check out the output files. First let's plot the XVG file with the statistics for different secondary structures of our protein. Particularly, it shows the number of residues belonging to some secondary structure element as a function of time. In the legend you can see all the secondary structure types which are supported by dssp tool. It has various applications, including protein folding, or when you want to compare two isotypes of the same protein, or when you want to study how some mutation impacts the secondary structure of some protein. You can use it to show that a particular part of trajectory corresponds to the equilibrium state of your protein, and you therefore can use those conformations for further analysis. Or if you know that your ligand bidding results in some significant conformational changes, you can use the dssp output to track down those changes and show them on the plot. The .dat file contains per frame data about secondary structures and residues belonging to them. Here each row represents frame, and each symbol specifies secondary structure, and the position of the symbol corresponds to the residue number which belongs to the secondary structure. You can focus on a specific part of this string. You can write your own scripts depending on your needs. It is fairly easy to analyze as the format is strict and explicit. That's all, folks. Thank you for your time, and let me know if you had any troubles.